Professor Gaurav Ahuja. So Professor Ahuja is associate professor at IIIT Delhi. He's a molecular biologist by training, but he's one of India's foremost experts in functional genomics. Professor Ahuja.
First of all, good morning to one and all. And my name is Gaurav. I am an associate professor at IIIT Delhi. We have a small lab in the center of Delhi, but a very beautiful campus. So I think many of you want to visit us, please come. So today I will be talking something, the work which we have been doing in our small lab at IIIT Delhi concerning aging. And in my lab, we try to understand how the chemical space works in the context of longevity. And so just wanted to show you some of the technologies which we have been building in the past few years in the lab. And for example, I'm very proud of this oncogen.ai because people are talking about LLMs and all that. We just helped the Rajiv Gandhi Cancer Hospital in New Delhi to build their fully automated AI-driven system from the sequencing till the clinical reporting, end to end. And the previous work, since they were relying on the iron torrent servers in the US in the queue, so the patient spends 3 days in ICU, the data analysis go on, and they look into the mutations, into surfing the databases, and then they recommend to the clinician which chemotherapy can go in. This procedure take around 10 to 15 days, but now it happens in 3 hours. Everything is automated. It is driven by knowledge graphs and generative AI. But today, since we are in a longevity space, so let me talk about two of the latest work we have got it from our lab. One is called AgeXtend, and another is called scCamAge. Now, just putting everything in context, so this is something I took it from my textbook, which I used to teach at the university. So in a classical programming, you normally have rules and data, and the classical programming try to find the answers. So, but with the invent of machine learning, we have data and answers, we get the rules. And we thought, why can't we use both of them? So the idea is, what are the rules in the longevity space and how can we find them? That is the simple question. So in order to do that, as a proof of concept, I just highlighted this because people try to identify carcinogens from the structure. And we basically, so one way to do that is you take non-carcinogens and non-carcinogens, you build models. But what we thought is, instead of, because if you see the numbers, there are only 2,000 carcinogens are available in the market. And the chemical space is 10 raised to the power 60. Irrespective, whatever algorithm you can use LLM whatsoever, you can never translate from 2000 to 10 raised to the power 60. So what is the strategy B? The logic which we use is, what a potential carcinogens can do in our body, it increases the chronic inflammation, oxidative stress, and so far and so on. So we modeled these pathways, and we haven't learned anything from the 2000 molecules, and we are able to predict all the 2000 correctly. So we put the years like this, not like this. So that's the strategy which we have used in this case. And this work was published in Nature Chemical Biology from the lab back in 2022. So now in the longevity space, when we were looking for the rules, and one of the beautiful things, I think the blessings in the longevity space we have is the hallmarks of aging. And if you see, this is the old cartoon. I started my longevity research with this cartoon, 2013. And one of the previous director from the Max Planck Institute, where I was a postdoc, Linda Patrich, was there. And now you see in 2023, there are multiple hallmarks keep on adding. So the more and more research has been invested in the last 100 years to build these hallmarks. Now the idea is, can we model each of these hallmarks and then build an AI model which can predict the pro-longevity molecules? That's the idea. So we did that thanks to these two beautiful girls in the lab, Ayushi and Sakshi. We got this AgeXtend build. I think December last year, it got published in Nature Aging, and I'm very proud of these two. So let me introduce you what is AgeXtend and how the logic it works. Now, AgeXtend is built, taken into account what the pharma requires, how we have to understand the anti-aging response. So AgeXtend has 4 different modules. We have the Garo prediction module, we have the explainability module, we have the toxicity module, we have the target module. So it starts with, you put any query molecule on the AgeXtend, it looks not into the chemical space, because normal chemists they look for the chemical similarities. It takes into account the chemistry, it looks into the targets, which proteins it can bind to, it looks into the activity, networks, and clinics all together. We call it as bioactivity descriptors, and then it says whether it is an anti-aging molecule or not. Not only this, if it is anti-aging, it moves to the explainability module where it also explains you in a biological sense why it is an anti-aging molecule. It can be altering your telomerase, it can be preventing your stem cell exhaustion, all the 9 hallmarks. Apart from that, it also predicts your toxicity of that molecule in the body. And last but not the least, as a scientist, data-driven science, it also tells you which proteins you should look into and do the ITC or the binding assays, which this molecule can bind to. Now, after just wanted to show you the performance, the AgeXtend works really nice. So we have got an AUCR of 0.9, and this is the testing data, 0.88, and very stable accuracy. You can see the scattering in the box plot. Moving a little bit more forward, people think it's fancy, but it's not. We have been compiling. So this is the DNA of AgeXtend. So we have taken the 100 years of knowledge about aging, which includes macroautophagy, which includes HSP inhibitors, NF-kappa B, sirtuins, everything, a total of 0.2 million molecules from the web, collated, collected from the last 3 years, and then this is the DNA of AgeXtend at the back end. And these are all experimentally validated molecules. It's a lot of work. So once we built that, what we did is, this is just a screenshot to show metformin is one of the nice tools. And the beautiful thing, when we were building AgeXtend, this paper came out, taurine and N-acetyltaurine. Both of these molecules, 3 together, predicted to be anti-aging. The beautiful thing is my model has never seen metformin, not even the nearby molecule, but still it can predict it is an anti-aging molecule. That's the beauty. And same for taurine and N-acetyltaurine. Moving a little bit more forward, so we have also taken one independent data set, which means all the, so AgeXtend was built on 2023 December. All the papers which came out on 2024 and 25, we took it as a testing data, and you can see nice prediction probability as close as near to 1. So we are able to predict all the anti-aging molecules which AgeXtend. We never saw them. That's the beauty. That is the rule-driven discovery. Moving a little bit more forward, this is the other work from the CamBL. We also predict very nicely the sirtuin targets, sirtuin 3 targets, and so far and so on. Moving a little bit more forward, this is something metformin, taurine, and acetyltaurine. We can predict very them nicely as anti-aging molecules, which is a proof of concept. The model can predict novel molecules as well, which is not the part of the training data. Now, is AgeXtend is biased towards anti-aging, or it can, so it also predicts the positive molecules. So we, as a negative control, we took DARCam, which are all those molecules which are inert. Like if you take 1 milligram or 1 kilogram of these molecules, nothing will happen to your body. We took that, and you can see the DARCam has a distribution like this, and the aging distribution is like this, very distinctive, which means AgeXtend is really specific to the anti-aging molecules. Now, after building this, I am very proudly making a claim. So we from India, from Delhi, we are able to contribute to the world a database of 1.1 billion compounds screened in Delhi, which includes all the molecules, Ayurvedic molecules from Chinese medicine, from the allopathic, from the PubChem, from the CamBL, everywhere we found a molecule. We calculated all the 4 modules. We contributed to the world as a database. So when people can see, if they have a molecule of interest, they can look into the database, they can find why it is anti-aging, and it has all the explainability. So that is the contribution we have made to the world. Now, what is AgeXtend is good for? So look at this. I mean, this is the CamBL. Earlier it was all gray. But now I have a tool which gives me a longitude and latitude, the GPS system. So now you can see this is a garoprotective chemical space where the probability of finding a novel anti-aging molecule is high. And this is what the space is given by AgeXtend. That's the beauty. We have not stopped here. So from my previous lab and also from others, people are more talking about microbiomes. People take the young microbiome, put it to the old in the form of FMT. So I have a very nice colleague, Tarini. So he has a longitudinal data of humans. So we have 5,388 humans longitudinal data. We have information about the age of the human, the gut microbes, and what metabolites they secrete in the host. Now, people often say this microbe is associated with longevity, this microbe is associated with longevity, but they never talk about what this microbe secretes in the body, because microbes themselves do not do the job. They secrete some microbial metabolites. So by using AgeXtend, we decode all the microbes irrespective of the microbial genomics data. We can actually find out all those species which are influencing the pro-longevity, and also what metabolites are responsible, which are produced by these species, which is responsible for pro-longevity. And this is done on 5,388 human data across 5 continents. We have also done that. Followed by that, we also tested some of the endogenous molecules because I always believe the body is the best chemistry. So if you do meditation, I think my previous colleagues talked about it. So the molecules, if you meditate, have a healthy life, you don't need, external pills. And many of the drugs you can see chemically, structurally, they are molecules, the endogenous metabolites. So we found out a lot of endogenous metabolites which are anti-aging. We tested them in the lab by using yeast, and you see there is a huge effect. Followed by that, we also tested senescence because one of the hallmark is the senescence. We also find out 2 metabolites which are anti-senescence, so senolytic in function. And last but not the least, we also help, perform some C. elegans with longevity assay. The beautiful part is, normally people say in machine learning, you take a molecule which is very similar to your testing data and look at the similarity. Similarity of these 3 molecules is almost equal to 0 with my training data. But still we have longevity response in 2 of them, not the third one. So which means the 2 molecules can be structurally divergent, but they can be functionally convergent. And that is something AgeXtend DNA is built on. It can predict that. Now, the next work, I just quickly, because of the time constraint, I just quickly pass through. This is the work which we have recently got it out. So Vishakha from the lab and Shubhadeep got this very nice scCamAge. My previous talk, EvoKG, they did it with humans. I did it with a single cell. The concept is same. You take a microscopic image, you get a single cell and try to ask the cell what is your age, how healthy you are, how much DNA damage you have, how good is your mitochondria, how much epigenetic alterations you have. All the hallmarks you can talk to the cell by using the image. That is something which we built. SC means single cell CamAge. So as you know, aging phenotypes are very constrained. So we have increase in size and all the other hallmarks are there. Very, very nicely, people have been accumulated from the last 100 years. So we ask a question, do genetically or pharmacologically alterations trigger the same type of longevity response? For example, I take metformin, I take resveratrol, I take rapamycin. Is the anti-aging waves what occurs in my body? Is this all the same patterns or they elicit at different times and different thresholds? That was the first question. Now the second question is, we ask, are aging-associated cellular morphofactory changes across the evolution? For example, I give you one example. One of my models, it has never seen humans at all. It is entirely trained on yeast cell. They look different, they behave different, but the only thing is people say, the death is inevitable, am I right? So the idea is aging is conserved. Now, if you build a model on the yeast data, yeast aging, and you project the human cell, is it going to predict the aging of the human cell? If it's, then the aging is evolutionary conserved at the morphometric parameters. So this is what we have tested. So just taking you very fast with this, I have few minutes. So what we did, we generated one of the largest data sets from the country, from Delhi, which is the chronological lifespan of the yeast from day 0 to the day 20. We built this algorithm which takes these single cells and it predicts the age, activity, explainability, and morphometry. Now, you can see this is a held out data, this is the predictions, this is the actual, so you can see very nice R square, which means the prediction and the core, there is a consensus between the prediction and the actual values. Moving a little bit more forward, the twist what we added, we make this, so from a bright field image, we make a coupled image, like the paired image, with the mitochondrial activity from the ROS levels, from the vacuoles, from the proteasome degradation and so on. So the idea is, you give me a bright field image, I can predict what is the mitochondrial dynamics has been happening. So these are the paired data. So this is the regression curves, which we have got for 3 activity. And this shows, this is nice because people have know when we age, the fattier the cells increase in volume, so we can nicely replicate in our scCamAge. So we do all the computation of the cellular surface, volume, and everything. Now, this is also explainable because it also pinpoints the subregions in the cell. This is a single cell. It also tells you why the model thinks it is day 2, day 10, day 20, and so on. Now, this is the actual test. So metformin is a magical molecule. I don't recommend to others to take it, but in the lab, whenever we throw metformin on multiple model system, it always works. It's a very good pill. Now you can see that we use metformin, spermine, and spermidine. These are all known anti-aging or pro-longevity molecules. This is my predictions, these are my cells. So if you see that the prediction says it is day 2, but the actual chronological age is day 10, day 12, day 16. So metformin takes everything to the back. So that is how you can use the single cell image. So you don't need to do the longevity assay for 20 days, 25 days. You stop your experiment in 5 days. You don't need to spend resources. You ask and predict the trajectory. I just need one more minute. And then once we did with the molecules, we also take a couple of already known pro-longevity and anti-longevity knockouts. We predicted scCamAge works perfectly. Moving a little bit more forward, one of the nice things which I learned in Max Planck is what doesn't kill you makes you strong. So one of the hypotheses is I keep on giving a stress to a cell, but not persistent, but in a, in Europe when I was there, so people used to take this, sauna and then they jump into the ice. So they do the, pulsative stress. So that is what we did. So we took the yeast, we flipped them into 30, 37, 30, 37, and so on for multiple generations. The idea was I can able to see longevity effect because they know the stress can come anytime, so they rather prepare their DNA, epigenome, proteome, and so on. But the bad thing is when we did this quantification, I could not see any difference. And I was just like sad, like we did it multiple times and we were sad. So people often say, what doesn't kill you makes you strong. What doesn't kill you also doesn't give longevity. At least this data was telling us. We were sad in the lab. Then in yeast, what we found out, yeast has 2 types of longevity response. One is chronological age, another is replicative aging. So when we looked into the replicative aging, it was wonderful. So you can see so many buds are there on the yeast, whereas in the case of pulsing with the temperature 36 and 37 and 30, we have a very significant low. And then we wanted to know what is happening at the molecular level. We did transcriptomics, metabolomics, integrative analysis. All the genes, what we found out is all known very nicely in the anti-aging response. So the idea is the pulsing temperature pulse gives you a longevity response at the replicative aging, not at the chronological age, at least in the context of yeast. My last thing, as I said before, so we built this data on yeast and just closing, sorry, just 1 minute. So we built this data on yeast, but then we project this data on the human system. We have got a very nice, thing. So it is without additional learning. So these are the original weights. And then when we relearn the system with 1 or 2 more neural layers, automatically the learning becomes enhanced. Sorry, I will discuss if somebody has questions because of the time. And yeah, so just so this is one of the largest data sets we have contributed to the world in the space of longevity from the lab. It has about 1 million single cells capturing the age, capturing the bioactivities. We put it for public as an open source resource in the longevity space from the lab. And it is available to everybody from our lab page. And with this, I think this is summary. I think, again, it's time's up. So this is important slide. So these are the funding agencies I would like to thank. So they supports my lab. And this is the beautiful team I have at IIIT Delhi. And these are all my collaborators. I want to thank Dibarka, Tarini, and Parul who are involved in this project. Thank you so much, and sorry for taking time.